Greetings to all the students of Medical Laboratory Technology UiTM. Today, we embark on an intriguing journey into the realms of blood typing procedures and serological diagnosis. We'll delve deep into the step-by-step process of determining ABO and Rh blood groups, as well as diagnosing infectious diseases like HIV, hepatitis, and syphilis. These procedures are crucial pillars of transfusion medicine and compatibility testing, shaping the very core of medical laboratory technology. Without further ado, let's dive into the fascinating world of blood typing. Blood typing is a crucial process in the field of transfusion medicine and compatibility testing. Now, you may be wondering, what exactly is blood typing? Well, it's the method we use to determine your blood group, and it's important in a variety of medical situations, most notably during blood transfusions. Picture this. You have two individuals. One needs a blood transfusion, and the other is a willing donor. The catch? We can't connect the two and start transferring blood. We first need to ensure that their blood types are compatible. This is where blood typing comes into play, so let's dive a little deeper. There are four main blood groups: A, B, AB, and O. And within these groups, we also have the Rh factor, which can be either positive or negative. And here's the thing, each blood group has certain antigens and antibodies. For instance, if you belong to the blood group A, you have A antigens and B antibodies. Now, why is this important? It's simple. If you transfuse the wrong blood type into someone, their immune system will react to the foreign antigens, causing a potentially dangerous immune response. That's why we need to match the blood types correctly. But how do we determine the blood type? Well, there are several methods, but two of the most common are the slide agglutination and microplate methods. The slide agglutination method involves mixing blood with specific antisera and observing for clumping, which indicates a positive reaction. On the other hand, the microplate method uses tiny wells where reactions occur, allowing for multiple tests to be conducted simultaneously. Remember, these methods aren't just about determining the blood type. They also play a crucial role in diagnosing certain diseases. Many infectious diseases such as HIV, hepatitis, and syphilis can be diagnosed through serological tests, which detect antibodies in the patient's blood. Armed with this knowledge, we can now explore how these tests are performed. So, are you ready to delve deeper into the fascinating world of blood typing and serological diagnosis? Let's get started. It's time to delve into the step-by-step procedures of blood typing. First, let's tackle the slide agglutination method. This is a simple yet effective technique that allows us to identify the ABO and Rh blood groups. To begin, we place a small drop of blood on a slide. Then we add an anti-A or anti-B serum to the blood. Depending on the blood type, the red blood cells will either agglutinate or clump together, or they won't. For example, if the blood reacts with the anti-A serum, it means that the blood type is type A or AB. If no reaction occurs, then we're likely dealing with type B or O blood. However, to confirm our results, we must perform the same test using the other serum. If the blood agglutinates with both anti-A and anti-B serums, it's type AB. If it doesn't react with either, it's type O. To determine the Rh factor, a similar test is done using an anti-D serum. Next, we have the microplate method, a more advanced technique that allows us to test multiple samples at once. This method involves adding blood samples to a microplate, followed by the addition of the anti-A, anti-B, and anti-D serums. The plate is then incubated, allowing for any potential agglutination to occur. After incubation, the plate is examined for agglutination. The presence or absence of agglutination in each well can help us determine the ABO and Rh blood types. Interpreting the results of these tests requires a keen eye and an understanding of the principles behind blood typing. Agglutination indicates the presence of the corresponding antigen in the blood. Thus, if agglutination occurs in the well with the anti-A serum, the blood type is either A or AB. Similarly, if agglutination occurs in the well with the anti-D serum, the blood is Rh positive. With these procedures, you can accurately determine a patient's blood type, ensuring safe and compatible transfusions. Moving on, let's tackle another vital aspect of laboratory technology: serological diagnosis. Serological tests are a cornerstone of modern medicine, playing a key role in the detection of infectious diseases. These tests, which analyze a patient's serum, can identify particular antibodies that the immune system produces in response to specific pathogens. Take HIV, hepatitis, and syphilis, for instance. These diseases, while vastly different, can all be detected through serology. Each test operates on distinct principles based on the disease's unique characteristics. For HIV, we look for antibodies against the virus. In hepatitis, we search for antigens or virus particles. And in syphilis, we examine the patient's serum for antibodies against the bacterium Treponema pallidum. Understanding these principles is essential to interpreting the results of these tests accurately. The ability to diagnose these diseases early and accurately can mean the difference between life and death. Now that we understand the principles, let's examine how these tests are conducted. Understanding the procedures of serological tests is crucial in diagnosing infectious diseases. These tests are designed to detect antibodies in the serum of a patient's blood. When our bodies encounter an infectious agent, they respond by producing specific antibodies to neutralize it. By identifying these antibodies, we can pinpoint the disease the patient is fighting. Let's start by discussing the procedure for diagnosing HIV. The initial screening test for HIV is an enzyme immunoassay, which detects antibodies to HIV. If this initial test is reactive, it is followed by a confirmatory test, such as a Western blot or an immunofluorescence assay. The confirmatory test detects specific proteins of the virus, confirming the presence of HIV. Next, we have the serological tests for hepatitis. Hepatitis B surface antigen test is the most commonly used initial screen for hepatitis B. If this test comes back positive, it indicates that the person is infectious and can spread the virus. To confirm a hepatitis B diagnosis, the hepatitis B core antibody test is used. On the other hand, for hepatitis C, an anti-HCV test is used to detect antibodies to the virus. Positive results are usually followed up with a HCV RNA test to confirm active infection. Lastly, the serological test for syphilis is conducted in 2 stages. The first stage involves a non-treponemal test, like the rapid plasma reagent test, which screens for the disease. If the test is reactive, a treponemal test like the T. pallidum particle agglutination assay is conducted for confirmation. Interpreting the results of these tests is just as important as conducting them. A reactive or positive result indicates the presence of the disease, while a non-reactive or negative result suggests its absence. However, it's essential to remember that false positives and negatives can occur, so these tests are usually part of a larger diagnostic process. With these procedures, you can accurately diagnose diseases and help patients receive the appropriate treatment. Knowledge of these tests and their interpretation is a vital tool in the arsenal of every healthcare professional. Today, we've journeyed through the procedures of blood typing and serological diagnosis. We've unpacked the intricacies of slide agglutination and microplate methods and delved into the significance of these tests in transfusion medicine and compatibility testing. We've also navigated the world of infectious disease diagnosis via serological tests, understanding their principles and interpretation. Remember, your role as a medical laboratory technologist is vital in ensuring patient safety and health. Keep learning, keep exploring. Thank you for joining us today.